what that is or what the extent is, and it's basically this green strip of rainforest that goes all the way from Alaska down to California. A really fascinating area because of the huge rainfall that happens and the massive transport of freshwater and the terrestrial materials to the coastal ocean. And what we're doing with this Coastal Rainforest Margins Research Network, which is an NSF-funded project, is we're really looking at, so my role is examining the marine impacts of these terrestrial outflows. Right, so that was my quick summary, and now I'm going to hand over to, starting with Haley, everyone else to tell you about their projects and themselves.
Hey everyone, I'm Haley, and as Brian mentioned, I'm working with him and Stephanie, and I'll also just mention that I have a couple other pretty key collaborators. One of them is Jennifer Jackson, who's at Psychiatrist in my office, and Charles Hanna at the Institute of Ocean Sciences, the IOF Lab. So I'm a physical oceanographer, and I've been focusing on the physical ocean properties. And what we'll be talking about right now is for Johnson Strait in particular, since that's a key leg of the salmon migration route for juvenile Fraser River sockeye. Okay, so I've got a map here of, let's see, is this going to do a laser pointer? Great. Map of the study area that I'm looking at for Johnson Strait. We've got the Strait of Georgia down here. This is Vancouver Island. Over here we've got Queen Charlotte Strait and Queen Charlotte Sound. And Johnson Strait is this very deep, narrow channel that connects Queen Charlotte Strait to the Strait of Georgia. There's also Discovery Passage over here, and I'll talk about that in a little bit. And so I'm primarily looking at CTD data, so temperature, salinity, dissolved oxygen, and also some nutrient data for very recent years. Since 2015, the Hakai Institute has been collecting data at a number of stations within Johnson Strait during the spring and summer months. There's a beautiful time series now for those years. From intermittently from the 1930s, particularly in the 1970s and 80s, and from about 2000 onwards, DFO has collected quite a bit of CTD data throughout this region, so I've got a ton of data to look at there. So I'll just show you an example of some of the data that I'm working with. This is from, just jump back for a second, from this little station right here, one of the Hakai stations, JS2, and we've got years, so this is 2015, 2016, 2017, and then the plots are by month on the bottom for potential temperature, salinity, potential density, and dissolved oxygen at the bottom there. And you can see that this is a really great time series, and it really helps me to look at the dynamics of Johnson Strait. So this area was studied quite a bit by Rick Thompson back in the 1970s and 80s, and the basic picture that he developed is over in this figure from one of his papers that I borrowed and added to. So we have dense salty water coming in from Queen Charlotte Sound. That water is usually as a result of upwelling, so it's very high in nutrients. And it's flowing over the sill and into the deep part of Johnson Strait. On the other side, in the Strait of Georgia, we have this freshwater outflow. So Brian mentioned that terrestrial freshwater, so especially in the spring, we get quite a bit of freshwater outflow coming in. And then in the middle of Johnson Strait, there's a really intense tidal mixing area. So this is where the physical oceanography becomes really interesting because we just have a ton of mixing. So we get quite a lot of modification of the water, and we create two new water types, a fresh pressure component going out at the top and a denser component going out at the bottom. So there's some question about the exchange happening through Johnson Strait because of that strong mixing. And in the time series, you can really see the competition between the salty, dense, very low oxygen water that's as a result of upwelling, and then the really near surface, fresh and light water, that's that runoff. And if you look at this spatially, you can see even more clearly that Johnson Strait really is very much a transition region. And so from the perspective of, say, a juvenile salmon entering this region, here's the near surface water temperature averaged over the top 20 meters. So each of these dots is one CTV profile, and this is both the Hakai data and the historic DFO data. And over here in the Strait of Georgia, temperatures are significantly higher than when you get into Johnson Strait and out into Queen Charlotte Strait. However, the other thing about Johnson Strait, we know it's very low productivity. We know that there's not a great deal of phytoplankton available for food for the zooplankton that then provide food for the salmon. So it's a pretty challenging area from a feeding perspective. And I think a major part of the reason for that is if we look at the near surface stratification, so this is just a buoyancy frequency measure of the stratification in the upper 20 meters, you can see that all along this Discovery Passage and Johnson Strait, we have this very, very big stratification. So there's nutrients there, there's sunlight, but the stratification is too weak to allow productivity. So it's a transition region because of that time frequency. And one last thing that I'll just mention that we're doing that is kind of interesting, to tie that piece of my work a little bit more into the upwelling and the nutrient availability. This spring and summer, we're going to be deploying an autonomous ocean glider, this guy right here. It's Stephanie Waterman's UBC ocean glider, and we'll be deploying it from the Hakai Field Station on Calvert Island, and it will travel across the shelf break to the coast, or across the shelf to the shelf break, and return to look at upwelling in these deep submarine troughs that we see, and to give us a better idea of how those nutrients will eventually reach Johnson Strait.
Hi everyone, so my name is Fernanda. I'm going to talk a little bit about my background and my postdoc project. I'm working with my Sierra Costa at Ubik and Brian Hunt through his collaboration with Hakai, as I mentioned before. And those are some of the members and collaborators. So my postdoc project is called the use of high-resolution satellite data to define coastal biogeochemical optical provinces. We are calling it as COBOPs, along British Columbia and Southeast Alaska. But first I'm going to talk, I'm a marine biologist, and my master's and my PhD was in biological oceanography. My main background is on phytoplankton and primary production, and I used to divide in three main research areas that I've been focused in the last couple of years. So I have done some ecology, and I call this biogeochemical process, which are some process that actually affects the ecology of the phytoplankton ecology. I have done some work with photophysiology and community structure of the phytoplankton. And another large part of my research is on biologists and remote sensing of the ocean color. They are actually three very related topics. So, for example, if you think on how the physiology affects the ecology of the marine process, then that's going to affect what's in the water, and finally it's going to affect the color of the surface of the ocean, and that's going to impact on the remote sensing work. So they are actually related, but now my focus for this postdoc is on biologics and remote sensing. So the project, the main goals of the project are using remote sensing data in high space and time resolution across British Columbia and Southeast Alaska to describe the variability in biogeochemical properties, such as chlorophyll A, total suspended matter, and dissolved organic matter along the terrestrial marine interface. Identify hotspots of primary production along this nearshore domain, and define what we are calling as COBOPs. So this is an image of the high-resolution sensor called Sentinel-3. And this is a nice image from 16th of March, about 10 days ago, for chlorophyll in the Strait of Georgia. And okay, and why it's important to use high-resolution data for this region. So as Brian was saying, this is a very complex nearshore domain. So we have many different processes happening at the same time, which creates different ecosystems and different landscapes and processes along the entire coast. So we have different discharge, different runoff source, and then we have fjords and estuaries. So it's important to they all happen in small scale, small spatial scale and time scale. So it's important to use high resolution data to be able to solve for this process. And the classification in provinces, they are useful to how to define the importance of each ecosystem unit along the coast, because they have different functions and ecosystem service. So, for example, some regions are more important for salmon migration habitat or for carbon sequestration and so on. The project has mainly 3 areas. I'm just going to mention 2 of them very briefly, how we do the calibration and the validation of the products from remote sensing. So the calibration of the sensor is basically the atmospheric correction that we have to do for the image. So that's the main focus of the FOCUS project. So the way it works is we have a sensor on board of the ferry. So this season, the sensor is installed at the Queen of Alberni, and this sensor is basically measuring the same signal that the satellite's measuring, but the sensor is measuring that without the influence of the atmosphere, the atmospheric signal, which we have to remove to be able to use the product. So this is one way we can do the calibration of the sensor. So we can investigate which model is the best to use for our region, because we know that this also changed for ocean area, for more offshore area, or for more coastal area, and so on. So that was very brief. Another part of the project is the products validation. So from the signal, well, I should have said that, but this is an example of the data from remote sensing reflectance, which is measured by this sensor and also by the satellite. And from this data, we use some algorithms to derive some products, such as chlorophyll, TSM, and CDON. So another part of this project is to validate how these properties are doing. So if you are lucky enough to have a very good image like this one on the 16th of March, and you were out there sampling, so you can actually sample for those in situ parameters, and then do filtration and all the lab work, and then validate, investigate how the products are doing, and then you can estimate for errors and uncertainties and wow. So I think that was just to make me motivated, because that was the first sampling of the season, and so that was just to make me excited to start. But that's very rare to happen, of course, to have a cloud-free and you can manage the forecast and be out there and, you know, the right time, and also have a phytoplankton bloom happening at the same time. So that was very nice. So I'm still working on this data, of course. That was, yeah, next time. So thank you.
Okay. So my name is Natalie Mahara. I'm a master's student here, working with Jenny and Brian. And if you've talked to me for more than like 30 seconds, you probably know that I love zooplankton, particularly zooplankton taxonomy. That's my jam. These are all pictures, mostly from the northern Strait of Georgia, that I got to take when I was playing with live samples from Quadra. So they're really beautiful and they're really diverse. And so the area that I'm interested in is the Discovery Islands and Johnson Strait region. So you might have gotten the idea already that it's a really dynamic region. You've got some areas that are really well mixed. You've got the waters turning, and you've got different levels of chlorophyll production. And then you've got other areas that stratify really nicely. In the spring and summer, you get massive spring blooms that you hear so much about. Kind of the tools that I use to answer our question of, well, how do zooplankton communities differ among this region? First, we collect the zoops. That's a jelly in my hand. I'm not just, that's kind of transparent. They're really pretty in the water, less so out of the water. Then you inspect the zoops, which meant about 10 months of sitting behind a microscope. Sam and Vanessa, I know they feel me a lot right now. So a lot of sitting behind a microscope, finding out what species, what stage structure and stuff is in the samples, and then assessing the zoops, which is lots of frustrating R code, which I think the vast majority of us can relate to. So that's kind of the 3 steps to my project. And so how do the communities differ over this range of mixing and productivity regimes in a really complex and dynamic system? You can answer that in a lot of ways depending on who you ask, and so the things that I'm most interested in are within the regions, how do they compare, how does the inter and intra-annual seasonality compare? So in some regions, like Johnson Strait, there's very little intra-annual seasonability with zooplankton. So the zoops that you see in like April, May, June, July, kind of the same all the way through, whereas in like the Strait of Georgia, there's this really, really clear seasonality. As well, what are the abundance and biomass dynamics like? So do you see a spring bloom, where that's the max amount of biomass, or is it different in different systems? Yes. As well as seasonal succession, do you see like zooplankton growing up from babies to adults, or are adults just getting swept in from somewhere else? Additionally, what are the key species and environmental parameters that drive differences between regions? And of course, what are the implications for charismatic megafauna, like baby salmon? So my study sites kind of span over the main routes that salmon can take. So it's quite a bit of information that you can get. So this is a scary looking figure. Don't worry about it too much. Basically what it is I counted the zoops, I put them in the computer, and then I said, computer, please tell me which communities, so what samples are the most similar to each other? And it spewed out this figure with lots of help from Yulia for coloring it appropriately. And so basically, you see that you've got this really beautiful, distinct Northern Strait of Georgia community mostly, Discovery Island site, and then Johnson Strait community is super, super different. And then one step further, for anyone that speaks Latin or likes zoops, I threw up some of the species that are driving those differences. So in the Northern Strait of Georgia, Tomopteris is a polychaete, pretty big polychaete, and you've got a lot of bigger copepods like Eucalinus and Neocalinus. Neocalinus used to be the dominating member of the zooplankton community biomass, not so much anymore. As well as krill, like euphausids, those are a lot more common in the Northern Strait, as well as they're more in the Northern Strait as opposed to just not really existing in the other samples. And then in the Discovery Islands and Johnson Strait, you've got lots of meroplankton. So those are the species that only spend a portion of their life cycle in the plankton, so things like barnacles, decapods, bivalves, things like that, as well as some smaller zoops. And in the Discovery Islands, you've got a lot more jellies and small copepods, whereas in Johnson Strait, these are just specific copepods and a decapod. Yeah, so that's just a little portion of the stuff that I found. And then we'll move on to Sam.
Great, so I'm kind of taking from one step higher up the trophic web from Natalie, and looking at what the juvenile salmon are eating in this area of their outmigration. So I'm asking, how do their diets vary, both spatially and temporally, across their outmigration through these two areas? And this is sort of what our field work looks like. We go out in a 19-foot aluminum boat, and we have our modified purse seine. We're able to target fish activity and pull them in, look at them, and we take samples of the 5 different Pacific salmon species as well as herring. So I'm going to be talking to you just about the the first part. So I said I'm looking at spatial and temporal things, but so far I'm just going to tell you a little bit about the spatial differences that I've seen. And to do that, we've got these 2 regions that you're getting to know very well today. So Discovery Islands and Johnson Strait, with 6 sites in each. And I'm just looking at a 1-week sort of interval, at the first week of June in 2015. And I'm taking 10 sockeye from each sample, as well as a zooplankton toe. And so from this, basically, I then do a very similar process to Natalie. We go out, we collect them, come back in the lab, spend months and months of your life over a microscope looking at what's in the stomachs, and then put it all into art and try and make some pretty pictures. So one of the measures I'm going to talk about today is gut fullness index, which is sort of a measure of feeding success for fish. And it's basically the weight of the stomach contents divided by the weight of the fish converted into a percentage. And so in the literature, generally juvenile salmon in the wild have a gut fullness index between 1 and 3%. And these site numbers won't mean anything to you along the bottom, but it's generally going from sort of northern Strait of Georgia, progressing further and further north, up to Queen Charlotte Strait on the right. And so when I put my data in, this is sort of what we see. And so I was originally expecting, we were talking about the unique oceanographic properties that you see in Johnstone Strait, making this potentially a food-limited area. Some have called it a trophic gauntlet for salmon, just due to that high mixing, high turnover means there's not a lot of food available. So I was expecting to see sort of a gradually decreasing trend like this. However, we found first a decreasing trend and then an increasing trend, which is not what I expected, but in hindsight does make a bit of sense. And so what we're seeing is sort of here, these are mixing areas where you've got the stratified waters of northern Strait of Georgia, or up here, Queen Charlotte Strait, that are being mixed with the highly mixed waters in between. So you see an increase in likely productivity and food availability for salmon. However, some of these sites in the middle, we are seeing very limited food availability, so potentially some starving conditions in these areas. And just to show you what those actually look like, this one would be this site here, and this is a sample from this site here. So,
can you explain what those are?
Sure. These little piles of goo are Ophioplura, or larvaceans. And down here, we have lots of copepods, or Matridia pacifica here, as well as some juvenile euphausids. These are some keyed gnats, and you have some amphipods as well, and lots of other different types of small copepods. So some of the diets are really diverse, some are not so diverse. And so, to tell you a little bit more about that, these are the same sites, sorry, I just numbered them differently, but going again from sort of northern Strait of Georgia to Queen Charlotte Strait, you can see right away there's some very clear differences between these two regions, which is really cool. So we've got Oikoplura and some more euphausids apparent in those stomachs. Again, the picture I showed you was this one here. And this is relative biomass of the stomach contents. So you can see in Johnstone Strait, they're largely dominated by these calamine copepods. And I also looked at the differences in sizes of the prey items. And so the sizes that we're seeing in Discovery Islands are actually quite a bit smaller. Most of the zooplankton are less than 2 millimeters. However, in Johnstone Strait, the majority of these calamine copepods are in the 2 to 5 millimeter range. So some next steps, I mentioned that we can also collect zooplankton samples, so I will be looking at those and being able to look at what zooplankton are in the water right at the same time that we're collecting the fish, and looking at what's in the stomach, we get a sense of how selective the juvenile salmon are being as they're feeding. I'm also going to be looking at the effects of dial feeding patterns. So when are salmon actually eating the most? Is it in the morning or in the afternoon? And how does that affect feeding or diet studies? I will also be doing a gut evacuation experiment, which is essentially how long it takes food to pass through the gut of the salmon. And then a temporal analysis, so looking at the diets across the entire outmigration and looking at it across 2 different years to tell us a little bit more.
Yeah, so I'm Jessica, I'm a postdoc with Brian, and I started my postdoc position last November. And before that I did my PhD in Germany at the EOMA Helmut Center for Oceanography, so my master and my PhD was mainly in biological oceanography. So now working with Brian, I changed a little bit from experimental ecology and food web ecology to, or added more the salmon perspective to that. And my topic is the spatial and temporal variation of juvenile sockeye condition and the growth rates of sockeye. So what I'm doing, again, you see the Discovery Island and Johnston Strait, so I try to compare between the Discovery Island area and the Johnston Strait area, and especially, or currently I'm looking between the years 2015 and 2016. So you see here the juvenile salmon migrate from the northern Strait of Georgia through the Discovery Island and Johnston Strait out of Sound. So what I do to get the condition and growth rate of the juvenile salmon, I quantify the nucleic acids and their ratios. So mainly I look on the RNA, the DNA, and the ratio of the RNA and DNA. So this provides a short-term measure of the condition and growth of juvenile salmon, so from the last 3 to 7 days. So which is kind of good because we have this high density of sampling locations, so I can really follow the fish between the years and the locations and if there are changes. So in theory, DNA concentration in a cell keeps constant, but the RNA concentration increases or decreases. So when we have a high RNA concentration, we have a high protein biosynthesis. This means we have a reasonably well-fed and metabolic active fish, whereas when we have low RNA concentration in the cell, it's not well-fed, it's low metabolic active and has a slow growth rate. So when we have a low RNA to DNA ratio, we have not such a good fed or in good condition salmon. So what I try to do in my project is I identify the drivers of these RNA DNA changes. So first I take the samples from the juvenile salmon and get the RNA and DNA ratios, which can be directly affected by temperature or salinity changes, but can also be affected by the food. So I also have later to look what's actually going on in the food sources. And this can be changes in the quantity, so the biomass or species composition of the biomass, but also in the quality, so species composition, but also the fatty acid content, so the quality of the food. But also, again, this can be affected by the phytoplankton, and this can be affected by environmental drivers again. So it's a huge thing. So currently I'm just analyzing the salmon. So the rest comes later. So I'm showing some early results. And I have 2 locations, which is DO9, which is in the Discovery Island, and DO7 and JO7, which is in the Johnston Strait. So here you can see the RNA-DNA ratio of both locations at 2 years. So the black color represents the year 2015, and blue represents 2016. And what we can actually see is that we had a lower RNA-DNA ratio in 2016 in both locations, but also a lower RNA and DNA at DO7 between 2015 and 2016. So it decreased in 2016. And then I looked, okay, why can this happen? So I calculated the yearly average of the temperature of both locations during the sampling time. And what I saw is that we had a higher temperature or average temperature in 2015 in the Discovery Island, but no really effect or temperature effect in the Johnston Strait, no big difference. So what does it mean? And this is like going on, and I will develop during the next weeks and months. So, but still I have to do more analysis of the RNA DNA. But which is really interesting that we have this RNA and DNA changes between the different locations, but also between both years. So I also said, okay, what is also affecting condition of the fish? So therefore we analyzed the fatty acid profile of the juvenile sockeye salmon. This is just an example how it kind of works, what affects food quality. On the left side you have like a general picture, like the total amounts of fatty acids that are measured in a fish. Then you have the saturated fatty acids and also unsaturated fatty acids, which include like essential fatty acids, which are taken up just by predation or by the food source. But you can also look on the ratio of specific essential fatty acids like DHA, EPA, and ARA, which are important for the development and the growth of the juvenile salmon. So when you see changes between years and between the different fatty acids, this gives an indication if also the condition of the salmon or the growth was affected between the different years. So just an indication, I think.
But I believe most of you don't know me, so I'm going to take a minute just to talk about me. I did my master's and my PhD in Spain, working with small banana fish ecology. Then I moved to South Africa, where I kept studying small pelagic fish, also using soluble isotopes and fatty acids, as well as I did in Barcelona. From there, I went up the trophic ladder. I went to study tropical tuna in the Indian Ocean, reproductive and trophic dynamics of skipjack and yellowfin tuna. And just before coming here, I was a postdoc at Stockholm University, where I was helping to update one ecopath model for the Baltic Sea. In particular, I was providing that data for the seal consumption on the commercially important fish species there. And then 2 months ago, I moved to Vancouver to start working as a MyTex fellow at Brian's lab, in partnership with the Pacific Salmon Foundation, and in very close collaboration with the DFO at the Hakan Institute. So, if I had to give a title for my project, it will be officially something like this. And I say officially because the focus of the Pacific Salmon Foundation project is on Chinook and coho salmon, but I will be looking as well at pink salmon, sockeye salmon, but also Pacific herring. And the first 2 objectives of the project will involve the description of the pathways and connections of the plankton components in the food web in the Strait of Georgia, and how these pathways relate to juvenile salmon and to herring. And the spatial and temporal variability of these pathways. And I will be focusing primarily, but not exclusively, on the zooplankton, because that's where the main prey for salmon juvenile, as we have seen from Sam's presentation, is. We already have a very good coverage, spatial coverage of the entire Strait of Georgia, mostly thanks to the DFO surveys that go back to 2008. We have samples at least once or twice a year since then. Plankton samples mostly, but we also have a lot of data and samples from the weekly surveys that are conducted in the northern Strait of Georgia by the people in the Hakan Institute. And right now we are finalizing with the preparation of all those samples, practically all of those samples already. And we're going to do stable isotopes analysis that we hope in the next couple of months will help us answer these questions, among others. Which food sources support the juvenile salmon in the Strait of Georgia, and whether these resources vary from the south to the north of the Strait of Georgia. The next objective of the project will be to develop plankton prey quality index based on the nutritional value of the main prey of the salmon. And that's important because, well, we see here differences in the energy content of the different potential prey for salmon, but it has been also detected a diet shift, in this case for sockeye juvenile in the Strait of Georgia. We see that it goes from highly copper pots were highly important in the 60s, but then basically disappeared after that from the diet. Sorry. But we also have seen that there is an increase since the beginning of the 21st century in the decapod, in the decapod library in the diet of the juvenile salmon, which happens to be the less energy rich prey for that. And the last objective objective of the project will be to provide data to help integrate a biochemical model to a food web model in the Salish Sea. And well, that's the future, so I won't say much about that. And thank you.
So I'm Jessica. I just finished my Bachelor of Science in Oceanography and Biology. I did my honors thesis with Brian and co-supervised by Dave Pakamov. And now I'm a prospective master's student. So my honors thesis and my master's project both work with jellyfish, which is probably why I got my own slide in the introduction, is because it's a little bit of a departure from what everyone else in the lab does. First I'll talk about the drone work that we just did. So I work out of the Calvert station for Quadra. A lot of the field work is done up there instead of the Quadra station that everyone else seems to work out of. So the drone, this is just some drone footage of us working in the field. So the drone sits above the boat and helps to position us within the jellyfish aggregate. I guess for those of you that aren't familiar with jellyfish, they tend to make these really big aggregates, so that's kind of important to the project. And so the drone flies above us, and then we sample it with these nets. We do some net tows, we count everything, we measure it, and while we're doing that, the drone takes pictures of the aggregate, which we can crop and to kind of like take out the effects of the coast and stuff, because it's kind of complicated in here. But once it's cropped, we run it through something called a k-means cluster analysis. So it kind of classifies pixels based on color differences. And then usually all the jellyfish end up in their own cluster, which we can extract. So that's all these white dots here. It's all the jellyfish. And then we overlay the cluster analysis with 1 meter squared quadrats to give us kind of like a percent cover in a meter squared. So we have something to compare to the biological data. So when we compare the two, they're not, the magnitudes aren't directly comparable because obviously they're not the same measurements, but the trends that we saw when we compared the net data and the drone data, this aggregate D at low tide was the highest out of both of them, just like B was the lowest, and then these two follow the same track. So this result was kind of what convinced us that we should probably publish our data, and the publication came out in January, so that was super exciting. Yeah, so following that, I have plans to start a master's project in the Hunt Lab. So again, for those of you that aren't familiar with jellyfish, they have this stage that everybody's really familiar with. It's called the Medusa Stage. The Medusa Stage lays these little tiny larvae which settle into benthic polyps, which are super, super small. So they have this phase that's super important because they do asexual reproduction and divide and divide and divide and produce these little ephyra, which grow into medusae. So there's two important stages, the Medusa stage, and then the less known polyp stage, which is hard to study because it's very small and not as big as the Medusa stage. So my master's questions are going to focus on what the prey source is for polyps and medusae of Aurelia, which are moon jellies. If you guys spend any time near the water, you've probably seen one. So polyps especially are really hard to study hard to do feeding analyses on because they're very small, and so their gut contents at a certain amount of time are just very, there's not a lot of it. So the second question is, so we're going to take these two things together and determine what the role of Aurelia is in the local food web, so that kind of ties into the bigger picture that everybody else focuses on. Our hypothesis is that these Aurelia are going to be non-selective generalist feeders in both the medusae and the polyp stage, because, I mean, they just happen upon food, they put whatever they can fit in their mouth, and that's what we expect is we're going to see. So we want to measure the medusae feeding with a molecular genetic analysis. I have no idea why this is bigger, that's okay. We're going to measure them both with a molecular genetic analysis. But in order to do the stable isotope analysis, we need a lot of biomass, and the polyps are, again, very small, so we won't be able to get enough biomass to do that. But molecular genetic analysis will give us a snapshot of the food that will be in their gut at a certain time, and then the stable isotope and fatty acid composition will give us a more comprehensive picture of the medusae's life history in terms of what they're eating.
I'm going to say a quick couple of words about Wade and Boris. So yeah, they're not here, so hopefully you'll get to hear more about their work in the coming months. But just to say that Wade is working on movements, habitat use, life history, and spatial dynamics of, initially he started doing this with herring, but he's now been working on salmon as well. And he uses particularly otolith microchemistry. So the fish are laying down calcium carbonates, otoliths, and the chemical composition of these otoliths reflects to a large extent the environment that they're in, can also be influenced by physiology, feeding, etc. But we're particularly interested in the environmental components and using this to be able to map the distributions of herring and salmon on the coast of BC, but then also beyond, in the case of salmon, into the North Pacific. And so with the herring, he's particularly looking at natal homing, so whether fish come back to the same areas to spawn. And we're also trying to resolve the question of how adult, or which environments adult herring are using on the BC coast, because this is a pretty important management question, and there have been some assumptions based on fairly sometimes inconclusive tagging data, and we feel that the microchemistry is a nice tool for really mapping out the distribution of herring on the coast. But for fish that we're sampling at spawning sites in particular. And then for salmon, he's been looking, initially he just started off by comparing two stocks, rivers inlets and Chilco, and he's looking at, we get a full chemical profile for the otoliths across the lifespan of the fish, and just looking at strontium and barium, which reflects salinity, we can already see that Chilco and rivers inlet sockeye are using very different areas of the North Pacific, and again, this is an important piece of information to have when you're trying to understand how salmon are using the offshore habitat, which we know so little about, and how changes in that offshore habitat, which are not equal in space and time, might be affecting different stocks. So that's Wade. And then Boris is working on, he's particularly working on salmon, and sockeye. He started off working on sockeye salmon, in fact he's still concentrating on that. And what he's trying to do is really hone in on this high seas phase of salmon life history. And so doing some similar work to Wade, but then also extracting information on the food webs that the salmon experience on the high seas. And he's doing this using stable isotopes. And we have two sources of stable isotopes. One is we're working with the Wekino up at Rivers Inlet. So they have there used to be the third largest sockeye salmon fishery in BC, collapsed in the 90s, and right now there's only a food fishery allowed. So we work with the Wekino to collect samples from the food fishery, which we then analyze for the health of the fish, and we measure carbon and nitrogen stable isotopes. And we've also accessed a scale archive from the DFO management program. So they go out every year and they sample fish that are coming to the spawning streams. They take scales just to age the fish, and they log all these in scale books. And we found that these go back in time, and all the way back to 1913. So we've created a long time series of historic carbon and nitrogen stable isotope distributions for this particular stock. Which helps us, we can use this to say, initially we were just looking at using it to say something about the food webs that these fish experience, and particularly where the productivity was changing. But subsequently, Boris has found out, well, there's this very strong relationship between carbon isotopes and C13 signature and temperature. And so he's now been able to use the carbon isotope signature of the returning fish to predict their distribution at sea in the sort of 6 to 12 month period prior to return. And I don't know if you can see this here, the red distribution, the isolines is indicating the distribution of the rivers and their sockeye quite a lot further south than we expected. We expected the fish to be distributed further north in the Pacific. So what he's doing now is he's getting access to scale archives all around the Pacific Rim, and he's going to be doing a similar type of analysis to try and place the location of different stocks around the North Pacific. The other thing he's doing is we've tapped into this North Pacific Continuous Plankton Recorder Service. We're taking plankton samples from this survey, which go back to 2001. We actually have all these data already, measuring isotopes of this trophic baseline, the plankton, and using this to build isoscapes for the North Pacific, which feeds back into the salmon data and helps us calibrate food chain length experiment that for the fish are being given here the pre-clinical experience, which is telling us more about productivity and transport efficiencies in the system. I'm sure you'll be hearing more about this in the coming months. And so yeah, that's it. That's the, I guess, the lab summary. Thanks everyone for coming, and just to let you know that I'm very happy, and I think I'm going to speak for the rest of the lab. Everyone's very happy to talk about their research at any time, so feel free to approach us and discuss our projects.